Sanger sequencing, developed in the 1970s, remained the standard genetic sequencing method into the early 21st century. It has now been supplanted by next-generation sequencing, which encompasses several techniques and technologies that more rapidly, accurately, and cost-effectively sequence a patient's genome for the purposes of diagnosis and treatment. Strategies range from targeted gene panels, used when a specific disease or group of diseases is suspected, to clinical exome sequencing, which targets almost all the protein coding genes, which is suitable for cases of greater diagnostic uncertainty, to whole genome sequencing, which yields a complete survey of a patient's genetic makeup, including all coding and non-coding genes. Each approach has its own benefits and drawbacks, and the choice of which to employ depends on several considerations, including cost. For example, a child is born with dysmorphic facial features and exhibits slight developmental delays in sitting up and walking, but otherwise seems healthy. At 18 months, he begins to suffer periodic uncontrolled seizures that are unresponsive to conventional anticonvulsant therapy. Pyridoxine-dependent epilepsy, or PDE, is considered. PDE is a treatable autosomal recessive disorder usually characterized by seizure onset in infancy. A trial of pyridoxine while measuring brain electrical activity, however, shows no benefit. Between this failed pyridoxine trial and the delayed onset of seizures, PDE becomes an unlikely diagnosis. By 6 years of age, the child has learning difficulties and hyperactivity. Continued episodic seizures lead his doctors to propose DNA sequence-based diagnostics to help refine their treatment approach. The doctors consider ordering a gene panel to sequence selected epilepsy-associated genes, which would test for a number of treatable types of epilepsy, including PDE. But they choose the more comprehensive clinical exome sequencing instead, in order to minimize the chances of missing any newly discovered epilepsy genes.